Hi students, today we are discussing about amino acids in that biomolecules chapter 12th standard. Amino acids. So already all 20 amino acids last class we are discussing. So 20 amino acids, in that essential amino acids what, non-essential amino acids what, we discussed already. Now structure of proteins. Structure of proteins. Structure of proteins. Okay, structure of proteins. How it will be is their structure in proteins. Same like how biomolecules in that glucose, fructose, galactose, lactose or maltose, how it is structures. Same like this is also having structure. So their glucosidic linkage is there in glucose, fructose and all. So polysaccharides or disaccharides. Here also some monomers combine to form a big polymer, that is alpha amino acids. So alpha amino acids. How it is polymer of alpha amino acids? So proteins are combined. How it will be combined? How it will be combined? Connected or combined with linkage. One linkage. That linkage we will call peptide linkage. One linkage. That linkage we will call peptide linkage or peptide bond. Peptide bond. So peptide bond or peptide linkage. Two mono amino acids combined to form a polypeptide bond. Combined to form a protein, big protein. So how it will be combined? Peptide linkage between this peptide linkage is the peptide linkage between amino acid group, that is nothing but COOH acid group of first compound or first amino acid and amino group of second amino acid. So where it is that linkage will be happen, so that I will explain. If it is the same amino acid or different amino acids also connecting with one bond, that bond is peptide bond. How it will be connected, I will write here. See, glycine, I will take an example. Glycine means, yesterday already one class we discussed NH2CH. Here R or H, glycine, no, already we take glycine example. Glycine this is glycine, COOH. Here COOH and then plus NH2CH COOH, here CH3. So this is alanine and glycine, GLY, glycine. So glycine and alanine will combine to form what compound? See here, to a loss of water molecule from where second NH I will write like this, NH2 no, so this H and here one OH minus water molecule from glycine and alanine will lose water molecule. We will get, without that water molecule we will write down no space, so I will write here. So how that compound will be? See, from this I am writing, see NH2CH, here also one H, otherwise CH2 also we will write, glycine, and C double bond O, N attached here, so NH and then CH, then CH3 and COOH. So now this is that linkage is there. What linkage? Peptide linkage. This is peptide linkage, AGE, linkage, peptide linkage will be formed. Now this name is glycine and alanine, no, two alanine molecule also will form polypeptide or peptide linkage protein, or two glycines also will form this, like same or different molecules, amino acids form a proteins or peptide linkage, that's polypeptide linkage. So this name is glacial alanine compound name. So this is a structure, this is peptide linkage of one compound. If one linkage is there, no, one linkage only, so two compounds will form one linkage, that we will call monopeptide linkage. One linkage only, no. If three compounds will form linkage, that we will call dipeptide linkage. Three compounds will form one compound and one link, two compounds have one link, and three compounds, 1 2 3, this is 1 2 3, three compounds have two linkages, that's why dipeptide. And if it is four compounds means tripeptide linkage or bond. Five compounds means tetrapeptide linkage. So like that, if more than 10 we will call polypeptide group, polypeptide molecule. If more than 10 compounds connect each other, more than 10 we will call polypeptide linkage. Here example of this one is insulin having 51 compounds, 51 molecules combined, 51 monomers combined, 51 amino acids combined to form a polypeptide molecule. So that is example insulin, you know, no, insulin. So mass of insulin or polypeptide molecules is 10,000 unified mass, or AMU, or unified mass. So mass of these polypeptide molecules minimum 10,000 units mass it will be having. So that only we will call polypeptide molecule. Okay, next insulin is a best example for polypeptide. So this is about peptide linkage between two molecules. Now we will discuss about proteins can be classified as classification. Proteins can be classified as on the basis of structure, on the basis of proteins proteins classified on the basis of their molecular shape, basis of their molecular shape, classified on the basis of their molecular shape. So based on molecular shape, two types is there, amma. One is molecular shape. First one is fibers proteins. One is fibers protein. So fibers proteins means what? So fibers proteins is when a polypeptide chain, polypeptide means, now you know, polypeptide compound or polypeptide compound having straight chain or chain structure, polypeptide compound having chain run parallel or have chain parallel structure. So polypeptide compound having chain parallel structure is known as fibrous protein. Example of that, hydrogen and sulfide bonds, where it is connection is there, bond connected with hydrogen and sulfide. Between hydrogen and sulfide bond will be present, so our connection will be present in hydrogen and sulfide in fibrous proteins. So fibrous proteins are insoluble in water. It is insoluble in water. Fibrous proteins are insoluble in water. Example of this one is silk. One is silk, second one is wool. So this is examples of that. Common name for that is common name is keratin. Keratin means keratin. This is a name. Keratin means silk, wool, like all will come example. Air also example. Air also having keratin, silk also having keratin, wool also having keratin. So this is fibrous proteins. So fibrous proteins, polypeptide compound having chain parallel structure, bond connected with, in that also some bonds between hydrogen and sulfide. So it is insoluble in water. Example is keratin, silk, wool and hair is the best example. Okay, next disulfide bonds between disulfide hydrogen comma disulfide. This is important point. Where is bond present in fibrous proteins means hydrogen and disulfide. This is about fibrous protein. Next one is globular protein. That is a spelling GLO globorous. GLOBUL ROUS. LOR, globular proteins. Globular proteins. So globular proteins are, it is a round shape, spherical, spherical shaped molecules. It is soluble in water. It is a soluble compound. Before one is insoluble in water. Next, insulin example, best example, insulin and insulin and albumin. Insulin and albumin is the best example for globular proteins. So fiber proteins and globular proteins is this is. So example of globular proteins is these two, insulin and albumin. Next, this is about two classification based on their molecular shape. Two proteins is there. So some examples also we discussed. Next, structure and shape. Structure and shape of the proteins. Structure and shape of the proteins. How can, after many studies, structure, how it is there? Structure, it is having four types of structure. One is primary. Second one is secondary. Third one is tertiary. Fourth one is quaternary. Okay, secondary proteins having, primary proteins having which structure? Based on structure, no, the structure and shape, this is proteins having polypeptide, polypeptide linkage, linear shape. Linear means linear, it is one another connect with like this line, linear shape. So that we will call primary peptides or primary proteins. That is only primary. It is with the sequence, with how it is linear with sequence structure. Sequence means one order is there, that is primary. So example we will tell, this is primary structure, not this is like this. This is primary structure. Secondary means it is having two types. One is alpha helix structure. Next, beta, alpha helix and beta plated sheet. So alpha helix means how it is there? Alpha helix means structure like this is there, alpha helix. Beta sheeted means, alpha helix structure like this is the secondary proteins structure, like this is there. In this alpha helix means it is having alpha helix alpha helix like this is there. So here carbon double bond O and hydrogen, if CH2 between hydrogen is there, no, this hydrogen and this oxygen between, and here one double bond O, or here one H, otherwise here C double bond O, here H between bond, that means hydrogen bondings present in helix, alpha helix structure. This is, so plated structure means it is like steps like plated, this is like plated structure. Here also hydrogen and oxygen between hydrogen bondings is there, hydrogen bondings. Due to the hydrogen bondings, that shape is like this is there, two types, helix and a plated sheet. So that is secondary, it is existing two different types. One is this one, alpha helix. Secondary proteins having two types of existing two types of molecules or two types of shapes, that is alpha helix and beta plated. Alpha helix means like this it will be there, beta plated means like this it will be there. So hydrogen bonding, both are having hydrogen bonding, where it is CO is RC double bond O, here carboxy, so here oxygen and amine NH2 group is there, no, here hydrogen oxygen between one bond will be forming, that bond we will call hydrogen bonding. This is secondary. Next tertiary means three-dimensional structure of three-dimensional structure we will call tertiary, due to what is that, how it will form three-dimensional, due to bonding. Which bonding? Hydrogen interaction between the R group, due to chain interaction between alkyl group. Alkyl group means what? In that compound, like this is amino acid NH2CH and COOH group, no, here one R is there. So this alkyl group interact with other compound hydrogen or other compound oxygen, whatever it, that alkyl group interaction is there, that is a tertiary three-dimensional structure due to the chain interaction between alkyl groups is known as tertiary. Now quaternary compound, so secondary and tertiary compounds is there, no, secondary I said like this structure, tertiary means like this structure. So this is amino acids, both are mixed in this, that is quaternary, more than one amino acid chain, more than one, not one means this is one chain, this is also one chain, more than one means like this. So more than one amino acid chain present in compound, that is quaternary amino acids. So this is about amino acids. Next class we will discuss enzymes. So in this class what we discussed, no, I will tell simply. First one is amino acids classified as two types. So amino acids, how it will form? Polypeptide group, polymer, alpha amino acids combined, alpha amino acid, one acid and another acid combined to form one big molecule, that is amino acid, polyamino acid or polypeptide. So it is connected with one link, that linkage we will call polypeptide, not polypeptide, peptide linkage. If many linkages is there, means one big molecule means more than 10 linkages is there, that is we will call polypeptide linkage. So one molecule and one more molecule between one link is only is there, that is monopeptide linkage or monopeptide. If two links between three molecules, that is dipeptide linkage. If three links between four molecules, that we will call tripeptide linkage or bond. So it is connected to join one and other amino acids between where it is connected one acid group and amino group between connection is there, that is we will call peptide linkage. That only. What is peptide linkage? Questions will come in exams. What is polypeptide or dipeptide or tripeptide, tetrapeptide? Tetrapeptide will form 5 monomers of amino acid. It will be form a 5 linkages, like you should tell. Next, based on the structure, it is proteins can be classified into two types. One is fibers proteins and globules proteins. Fiber protein example insulin. It is insoluble in water. Insoluble in, not insulin, it is keratin. It is insoluble in water. Keratin is insoluble in water. It is having hair, human being hair also keratin is there, and wool and silk also having keratin. So it is insoluble. Next one, globular proteins. It is a round shaped polymer. Example, it is insulin and albumin. So it is soluble in albumin and insulin, also soluble in water. Soluble in water. Okay, next, structure based on some types is there. That is primary, secondary, tertiary. Primary having polypeptide linkage linear with a sequence. One sequence is there, means that is primary. So secondary means not a one sequence. Secondary having two types of structures possible, that is alpha and beta. Alpha 1, alpha helic means, you know, like DNA, how it is there, like that, alpha helic. So here one group, here one group connect with hydrogen bonding, that is alpha helic structure. So that is known as alpha helic structure. Now one more structure is there, that is we will call like steps, like that is beta plated. Here one, like hydrogen bondings between them, that we will call beta linkage. Okay, next, tertiary compound. Tertiary compound, three-dimensional structure due to the chain interaction between the alkyl group. Alkyl group means this R in this amino acid. Alkyl group is there, no? R means alkyl group. That R will linked with other compound, that we will call tertiary amines. Next, quaternary amines. Secondary and tertiary combined, that is quaternary. So more than one amino acid present in amino acid chain or amino acid group present in this one is we will call quaternary. So easy only. Thank you. Next class we will discuss about enzymes. Okay.